Bismillahirrahmanirrahim. Let's start our today's class for the preparation of scientific officer of plant breeding and genetics. Our class number is 14 and question types are MCQs. This class is managed by EPSA. Let's move towards our first question. Our first question is that collection of clone DNA fragments that includes all or part of the genome of a species is called as gene bank, gene library, gene pool, or karyotype, and the correct one is a gene library. A collection of cloned DNA fragments that includes all or a part of the genome of a species is called as a gene library of that species. Next question is that the chromosomes in the complement other than the sex chromosome are termed as antisomes, autosomes, allosomes, or anosomes, and the correct one is the autosomes. Two are the in case of human beings, two are the sex chromosomes, and other 44 are the autosomes. And this the chromosomes in the complement other than the sex chromosomes are termed as the autosomes. Our next question is that the part of the chromosome which is responsible for chromosome movement is called as centromere, telomere, or centrioles, and the correct one is a centromere. The part of the chromosome which is responsible for chromosome movement is called as centromere. Next question is that biparental crosses does not provide information about additive variants, dominance variants, epistatic variants, or all of these. And the correct one is a epistatic variants. Biparental cross does not provide information about the epistatic variants. Our next question is that which of the following designs are like line X tester cross? NCD1, NCD2, NCD3, or none of these, and the correct one is a NCD2. NCD2 design are like line X tester cross. Next question is that which of the following design is least powerful? NCD1, NCD2, NCD3, or none of these, and the correct one is a NCD1. NCD1 is least powerful design. A substance or agent capable of producing cancer is called as mutation agent, antigen, carcinogen, or mutant. And the correct one is a carcinogen. Carcinogen is a substance that is capable of producing cancer. Our next question is that when a particular locus of X chromosome is also present on Y chromosome, the situation is called as sex-limited, complete sex linkage, sex influence, or incomplete sex linkage. And the correct one is a incomplete sex linkage. When a particular locus of X chromosome is present on Y chromosome, the situation is called incomplete sex linkage. The next question is that a population of genetically identical cells is called as clone, callus, colony, or culture, and the correct one is a clone. Clone is a population of genetically identical cells. Our next question is that lysynchronism was resurgence of neo-Darwinism, Darwinism, Lamarckism, or anti-Darwinism, and the correct one is a Lamarckism. Lysynchronism was the resurgence of the Lamarckism. Our next question is that blood groups in the man is an example of epistasis, multiple allelism, isoallelism, or compound loci. The correct one is a multiple allelism. Blood group in the human is an example of multiple allelism. Our next question is that the characters studied by the Mendel are all are dash traits, dominant traits, quantitative traits, qualitative traits, or recessive traits. And the correct one is the qualitative traits. Mendel's characters which are studied by the Mendel are completely qualitative traits. Our next question is that quantitative genes are controlled by dash genes more or less equal in effect. Few genes, two genes, several genes are single genes. And the correct one is a several genes. Quantitative genes are controlled by several genes more or less equal in effect. In next question, dash is the tendency of genes to enter the gametes in combination other than the parental, linkage, crossing over, variation, or none of these. And the correct one is a crossing over. Crossing over is the tendency of genes to enter the gametes in combination other than the parental. Our next question is that which of the following designs are not influenced by maternal effects? NCD1, NCD2, NCD3, or all of these? And the fifth one is a NCD3. NCD3 design is not influenced by the maternal effect. Our next question is that which of the following designs involves F2, P1, and P2 population? NCD1, NCD2, NCD3 or none of these, and the correct one is the NCD3. NCD3 design involves F2, parent 1 and parent 2 populations. The NCD3 involves F2, P1 and P2, BC1 and BC2, none of these, and the correct one is a F2, P1 and P2. NCD3 involves F2, P1 and P2. Next question is that from the genotype capital A, small a, capital B, capital C, small c, capital D, capital D. How many types of gamete would be produced? 2, 4, 16 or 8, and the correct one is a 8. In a given genotypic situation, 4 types of gametes are produced. Our next question is that if certain loci are present in a single dose, such as X-linked gene, the condition is called homozygous, homozygous, hemizygous, or heterozygous. And the correct one is a hemizygous. If certain loci are present in a single dose, such as X-linked genes, the condition is called hemizygous condition. Next question is that any gene whose locus is on Y chromosome is called Y-linked, polydendric, holocentric, or sex-linked, and the correct one is a polydendric. Any gene whose locus is on Y chromosome is called as polydendric. Our next question is that a structural part of the centromere into which spindle fibers insert is called centriole, centrosome, kinetochore, or telomeres, and the correct one is a kinetochore. Structural part of the chromosome into which spindle fibers insert are called kinetochore. Next question is that a cross which involves parents different in single traits, monohybrid, dihybrid, trihybrid, or cybrid, and the correct one is a monohybrid. In the cross in which parents parents which involves a single trait is known as monohybrid. TTC provides information about additive variance, dominance variance, presence or absence of the epistasis, all of these, and the correct one is all of these. TTC actually provides information about the additive variance, dominance variance, presence or of the absence of the epistasis. All these factors are covered by TTC. Next question, the detection of linkage is possible when more than dash genes are considered. 3, 2, 1, or 4, and the correct one is a 1. Detection of linkage is possible when more than 1 genes are considered. Our next question is that the most convenient and efficient method to determine the strength of linkages to make back cross, test cross, hybrid, or cybrid, and the correct one is a test cross. The most convenient and efficient method to determine the strength of linkages is to make a test cross. Next question is that in human, out of 46 chromosomes, dash are the autosomes 46, 42, 44, or 40, and the correct one is a 44. 44 are autosomes and Two chromosomes are the sex chromosomes. That's why here the correct option is a 44. TTC is an extension of NCD1, NCD2, NCD3 or none of these. And the correct one is a NCD3. TTC is an extension of NCD3 pattern. How many, how any population are involved in TTC analysis? 1, 2, 3 or 4, and the correct one is a 3. Three populations are involved in TTC analysis. Our next question is that a dense body in the nucleus associated with the processing of ribosomes is called nucleosome, nucleoside, nucleon, or nucleolus. And the correct one is a nucleolus. A dense body in the nucleus associated with the processing of ribosome is called nucleolus. Our next question is that a replicon that may exist in a cell independently of the chromosome is called plasmid, hybrid, or plastome. And the correct one is a plasmid. Plasmid are replicon that may exist in the cell independently of the chromosome is called plasmid.